So today we're going to talk about the DNA mismatch repair mechanism. So before everything started, we have a parent strand, which is written here. And you can see that all the base pairs, as long as they are GATC, then the A base pairs are methylated as the epigenetic modifications. So after the daughter strand are synthesized during our previous lectures, we learned that it will not be methylated for a period of time. And now you can also see that this T right here, it is mismatched because we have the C on parent strand, and the T should have been a G, but now it's a T, so there's an issue. So because the two base pairs are mismatched, then that means this part of the DNA double strand are not as stable as the other parts. So that's when we have three proteins to introduce. The mut-S dimer, it functions as a dimer. It is called the mismatch recognition protein. And then there's mut-L and mut-H. Mut-L is also dimer, and mut-H has the DNA endonuclease activities. So once there are a mismatch happening, then the mut-S will first recognize this side, and then it will bind to it. And following that, the mut-L will also bind to the mut-S, and then it will also gather mut-H to bind to mut-L. So this is the preparation of the DNA mismatch repair mechanism. So after the preparation is done, the mut-H will begin to search for the methylated adenine in a GATC sequence. So because mut-H is an DNA endonuclease, then the double strand will actually fold like this, and then but this three protein which bind to this TC mismatch side will not move. It's just the strand will move, and then when they move this way, then the mut-H, this mut-H, will find and recognize the methyl group on the adenine. And then it will actually cut the daughter strand. The reason why it can recognize its daughter strand versus parent strand is that, as we mentioned before, the daughter strand has a period of time when it is not methylated. So after mut-H recognizes the A in the parent side, the parent strand, then it will cut daughter strand at the T next to the A. After the daughter strand is cut, we can see that there still cannot be any fixing happening because the two DNA strands are still linked to each other. So this is when a helicase called the UVRD is introduced into the story. And at this point, we can just ignore these three proteins. And the UVRD helicase 2 will just separate the double strand just right past the mismatch site. So maybe they'll just unwind until the CA. After that, we have a exonuclease digesting from this part. So it will digest until it also passed the CA. So all these parts of the DNA strand are gone. They are deleted by the exonuclease. And now, the last part is that the DNA pol 3 will come in, and then it will synthesize another again, the strand that we just deleted, and this time it is going to be correct. If it is not correct, then it will just have the whole mismatch repair mechanism will just happen again. And after the new strand is synthesized, then we have the DNA ligase creating a phosphodiester bond to seal the gap. So this is the whole story of the DNA mismatch repair mechanism.